Hi everybody, welcome to another Chats with Childry. I hope you're all safe and well. Sun is shining outside my window in Surrey, and I'm delighted to be joined by Matt Boggess. I never checked how to pronounce your surname actually, Matt, before we did this call, who's staff QC engineer and scientist at Patheon, Derek Wood, who's senior principal scientist at PPD, and Xiaochun Yu, who is also a senior principal scientist at PPD. And today we are going to be talking about the practical considerations of implementing extractables and leachable studies in a modern laboratory. And so I'm delighted to be joined by two companies who are part of Thermo Fisher Scientific to just talk about E&L and some of the challenges around that. So before we start talking about the topic specifically, Matt, would you mind just doing a quick intro on yourself and a little bit about Patheon as well?
Yes, my name is Matt Boggess. I am a staff QC scientist engineer at Patheon, which is located in Greenville, North Carolina. We were purchased by Thermo Fisher. I believe it's been 7 years ago now. We are the contract manufacturing site, so we are performing the risk assessments on several different products for several different clients that come to us for commercial manufacturing.
Brilliant. Derek, same for you, please.
Yes, hi, my name's Derek Wood. I've held a variety of roles actually at PPD for the last 23 years or so that I've been here. As I mentioned, PPD, we are part of Thermo Fisher Scientific as well. PPD is the clinical research group of Thermo, and our site where we have a majority of our E&L testing in Middleton, Wisconsin, is it supports basically a lot of the analytical laboratory test services for the company and overall. So I'm currently a senior business manager within my current role within PPD, but as I mentioned, I've had several different roles and functions throughout my tenure at PPD.
Brilliant. And Xiaochun, same for you, without obviously having to go through who PPD are.
Thanks. Yeah, I'm also, from the same side, from Middleton, Wisconsin, same side as Derek Wood. So I'm a senior principal scientist working at PPD for over 22 years, and I'm the technical lead, so working in the extractable leachable area for over 20 years.
Fantastic. Right. Now, my first question is actually addressed to Matt. And viewer, just so you know, the reason Matt's holding his phone to his ear rather than a headset, he's had a few issues trying to get connected to Zoom today. So he's been kind enough to carry on with this interview, but through his phone. So I appreciate you doing that, Matt. Now, my first question, though, is for those new to the topic of extractables and leachables, can you give us a brief definition as to why they matter?
Sure. So extractables and leachables are impurities that can potentially arrive from the container closure, product administration, and also product contact with manufacturing process equipment during the drug manufacturing process. These are most commonly residual organic compounds such as plastic monomers, additives, degradants, residual solvents, as well as elemental impurities, such as trace metals. The assessment of these matters due to the risk of product quality and safety due to the toxicity level of some of these impurities that can be pulled off of the container closure system or the equipment. And these could interact with the active ingredient in the drug product and cause toxicological effects.
Right. Okay. So let's talk about risk assessments. So why are risk assessments so critical in supporting new product registrations? I mean, what is the driver for ensuring the risk assessments are performed?
So the 21 CFR Part 211 does have two subsections that clearly state, one of which is subsection 65. Equipment shall be constructed so that surfaces that contact components, in-process materials, or drug products shall not be reactive, additive, or absorptive so as to alter the safety, identity, strength, quality, or purity of the drug product beyond the official or other established requirements. Then there's subsection 94 of Part 211 that states drug product containers and closures shall not be reactive, additive, or absorptive so as to alter the safety, identity, strength, quality, and purity of the drug beyond the official or established requirements. So these risk assessments, abbreviated as PERLs, P-E-R-L, process equipment related leachables, assist in identifying these problematic and possibly high-risk components. And if these assessments are properly executed, they provide a high level of confidence that the process train and the container closure system do not alter the drug product in a manner that could be clinically significant.
Right. Okay. Well, thanks for that. So what is the most challenging aspect of executing a PERL risk assessment?
So the first thing is understanding the details of the manufacturing process and how the individual components in that process are impacted. And this includes parameters such as exposed surface area, temperature, and exposure time during the manufacture of the drug product.
Right.
So all have the potential for exacerbating the mitigation of organic and elementable leachables from the polymer matrix.
Right. Okay. And how do you determine which components are in scope for a PERL risk assessment?
So components are considered in scope if they have product contact with the drug product during that process train. So the agency continues to provide piecemeal guidance relative, but are not specifically clear in specifying what should be considered in scope. So it has to be based on product contact.
Right. Okay. So from your perspective, though, what is your most common frustration then when attempting to execute a PERL risk assessment?
So risk assessment is essentially based on real data that's available, and for materials and components, that data early in the process needs to be available from each respective vendor. So there are some vendors, many do have this data, and many do follow common guidelines such as the BPOG and the ICH guidelines, and some do not, or they do not. The testing wasn't done under recently approved guidelines, so those components often have to be identified as high risk in the risk assessment based on the lack of that information.
Right. Okay. Now, I understand that you propose a two-pronged approach for executing the PERL risk assessment. So one targeting organics and a second for elemental. So why do you feel this is more efficient, or what is the motivation for doing that?
Well, it's really in a lot of ways a cleaner approach because the organic extractables and leachables are really just a separate class of impurities than the elemental trace metal impurities. And they're also governed by some different documents. For example, elemental impurities do have an official guideline from the ICH, which is Q3D,
Right.
As well as corresponding USP Chapter 232. And that document specifically outlines different options in addressing those elemental impurities in your assessment and moving forward with your drug product. Organics, on the other hand, as of now, do not have a clear, well-defined regulatory path. It's very suggestive and subjective. There are associated USP chapters 1663, 1664, as well as 1665 for handling plastics. But they do not carry the same weight as those addressing the elemental impurities.
Right. All right. Well, Matt, thanks for that. So, Derek, we're going to move over to you there now, and I'm going to ask you, but after the risk assessment is completed and at-risk components designated, what are the next steps?
The next step is to conduct analytical testing for the designated higher risk components for the PERLs, the PERLs from the manufacturing process, to generate and evaluate actual extractables data.
Right. And how is such an evaluation conducted then?
Well, there are compendial chapters and industry best practice group guidance that provide direction on this. These include USP 1665 and related USP 665, and also industry guidance documents such as the Bioform Operations Group, or BPOG, Best Practices Guide for Extractables Testing of Polymeric Single-Use Components used in biopharmaceutical manufacturing. 1665 is a general chapter for the characterization and qualification of plastic components and systems used in manufacturing pharmaceutical drug products and biopharmaceutical drug substances and products. And 665, which is to be official, I believe, in May of 2026, describes a full extractable study utilizing multiple solvents to evaluate higher risk components by exposing the components to model solvents used or related to the actual specified conditions of the process.
Right.
So just an example for aqueous-based products, which, of course, is most applicable, for example, for parenterally administered drugs, solvents, model solvents such as ethanol water or IPA water, and low and high pH buffers may be used to separately extract the components of interest via exposure studies, where the component is incubated with the solvent using agitation, such as shaking or mixing at elevated temperature and for a specified duration. USP 665 specifies 40 degrees C for durations of either 1, 7, or 21 days, based on the actual type of component and expected duration of exposure in the actual manufacturing process. And then there are longer-term duration extractions that are more applicable for items such as bags, bottles, their closures, certain tubing attached to containers, for example, also, and other such components that are generally used to hold samples for longer time periods, such as, and those would use the recommended 21 days exposure, for example, at 40C. Just to continue a little bit on in a little more detail, if that's okay, that I just wanted to note that the extracts and extraction blanks or controls then are analyzed using analytical techniques consistent with other reference compendial chapters. Matt noted USP 1663, for example, to screen for volatile, semi-volatile, and non-volatile extractables, and elemental extractables as well, utilizing techniques such as headspace GC-MS, direct injection GC-MS, LC-MS, and ICP-MS instrumentation. I did mention BPOG earlier. Just to note, the BPOG best practices document was compiled primarily through a group of pharmaceutical company representatives to address extractables testing of polymeric single-use components used in biopharmaceutical manufacturing, very similar to USP 1665. But the BPOG is more of an industry consortium based recommendation and a testing protocol. And though, although it's becoming more aligned with 1665 in later versions, it still includes some differences, a little bit more extensive testing compared to 1665 due to the inclusion of additional extraction solvents and multiple time points for certain component types.
Right. Okay.
Yeah, just a note too, about simulation leachable studies. So the primary focus of 1665, 665, and BPOG, everything I've mentioned, is around the extractables, emphasis of extractables or potential leachables. When we're talking about leachables, we're actually looking at the drug product. What impurities in the form of that can migrate, in the form of leachables into the drug product itself. So if there's a specific drug product being evaluated for the manufacturing process, then another step may be recommended. And that includes conducting a leachable simulation study where the drug product or process stream solution, the solution that will come in contact with those manufacturing components we discussed, are evaluated for potential leachables contribution by performing laboratory-based studies, simulating the worst-case temperature and duration of exposure from the actual process, and then analyzing the prepared exposure solutions versus proper controls, of course. These solutions are then analyzed again by those same techniques I mentioned before, the headspace, GC-MS, direct injection, GC-MS, LC-MS, and ICP-MS.
Right. Well, you just mentioned the analytical evaluation using the headspace, GC-MS, direct injection, GC-MS, LC-UV-NMS, and ICP-MS. There's
It's
a lot of MSs there, yeah. So what are the key aspects of the methodologies that are used?
Well, the key purpose for these procedures are to screen for a broad range of potential extractable and leachable compounds. You're generally not looking for any or very many specific targeted compounds. You're looking for a broad screening type application, but also one that will provide some semi-quantitative information that's relevant to your process and the components and materials being evaluated. So we do utilize a variety of detection modes and techniques and different method types to accomplish that broad screening capability. Different methods are used, as we mentioned, those different techniques. So, different classes of compounds are covered based on their volatility. And then, of course, also the elemental impurities is covered by the ICP. At PPD, we have developed these mass spec screening methods to detect compounds across a broad mass range. The GC oven program, for example, or LC mobile phase gradient and column selection, detector setup, and sample prep have been optimized for specificity and required sensitivity to the process to be, most comprehensive in detecting and being able to report, at least to a semi-quantitative standpoint, on these types of compounds.
Right. Okay. Now, Xiaochun, you've been very patient while the others have been telling me all about their expertise. So over to you now. So can you talk a little bit about the Extractables and Leachables study for the manufacturing components and how this differs to container closure components?
Yeah, so the extractable leachable for the manufacturing components. So for the extractable part, so they are very kind of similar, so extractable or for the manufacturing component to the container closure component. Both studies, so the components are extracted with different solvent. And so, for example, for parenteral drug products, the aqueous products, usually the low pH, high pH water, and there's some ethanol or IPA water mixtures. And then the extracts are screened for potential for extractables with different methods, like the headspace GC-MS, direct injection GC-MS, LC-MS, ICP-MS. So that's targeting volatile, semi-volatile, non-volatile, and elemental extractables. So the study both are guided by the safety concern threshold and analytical evaluation threshold.
Right.
So,
okay.
But there are some differences. So, like manufacturing components, they are usually a lot more different polymer material and components compared to the container closure components. And also they may be several different manufacturing solutions instead of just the final drug product solution for the container closure components.
Yep.
And there's one big difference also. So for the leachable study, so for the container closure components, so the drug product are stored in the container closure components. So it's easy for you to kind of set up different orientations, different temperature, and test at different time point for leachables. But for manufacturing components, for example, like the filling lines, so the drug product solution you have in the container closure component system. So you may not know if they are collected with a new filling line, and if they are collected at the beginning, middle, or later stage of the filling process. So it's usually also not practical just to set up a new filling line in a manufacturing facility to collect the solution for each, at the beginning, middle, and the latest phase of the filling process for each testing. So in many cases, for the manufacturing components leachable study, a more practical way is testing leachable for the manufacturing component by simulating the exposure of the manufacturing process in the lab. So like solution, you have the extractor, you have the manufacturing components, and then have the manufacturing solution just prepared in the lab, simulating worst case or worst case exposure of the solution to the manufacturing component, and use that solution for testing leachables.
Right. Okay. So you mentioned that the extractable leachable study is also guided by the safety concern threshold and analytical evaluation threshold. But some of the manufacturing components are not exposed to the final product solutions. How do you calculate the AET?
Good question. So the AET calculation is still based on the safety concern threshold, and it will be the final drug product dosage So, but you need a good understanding of the correlation, of the volume correlation of the manufacturing solution to the final drug product solution. So you need to consider, any dilution factor.
Right. Okay. So what are the main challenges?
The final drug product solution.
Sorry, I don't realize. Sorry. So my next question: what are the main challenges for the extractables and leachable study for manufacturing components?
Well, yeah, for the challenging part, so for like extractable study for the container closure components. So the biggest challenge is probably identifying the extractables. So typically, so you expect to see many extractables from the manufacturing component with amount at or above AET level. So identifying those extractables is probably the most of the challenging part of the study, which will require kind of extensive knowledge and experience in mass spectroscopy and the polymer chemistry, and also good understanding of the manufacturing process. And that also requires some expensive equipment like a highly stringent LCMS and highly stringent GCMS. So without those identification, so it will be very difficult to make a safety assessment of the extractable from the manufactured components. So another challenge is it's kind of unique to the manufacturing components that it's hard to get the representative material for some of the study. So some manufacturing components, like some of the bag or bottle, they're 1,000-liter bags or bottles. So it's hard to do the extraction or especially the simulation extractable leachable use that large bag or bottles in the lab. So you can, in those cases, so you may use, scale down size, the bag or bottles, but the question is, it's also, if the scale down size bottles or bags, if that can be truly represent the extractive profile of the larger size bags or bottles.
All right.
And for the common, yeah, go ahead.
No, sorry, I was interrupting you. Finish what you're going to say. Sorry.
So for the common problem for the extractable leachable study for the manufacturing components, the first common problem we are seeing is analytical method that are not able to reach the AET level. So because like the USP or BPAC, they specify the extraction volume and the surface area ratio. So if you test the solution as is, that in many cases, so and then you'll probably do that way. But the method for some large dosage drug projects, so the method using the solution as is may not have the sensitivity to reach to the AET level. So that's one of the common problems. So in this case, you need to kind of concentrate on the solution first to make sure the method can reach to the AET level.
Right. Thank you, Xiaochun.
Another common problem. Another common problem.
Yeah, you were saying there's another common problem.
So,
no, please add. If you've got one more common problem,
another
please.
Common problem with another common problem with the unknown peaks. That's like I said, there are many extractable and some it's going to be hard to identify. So that one common problem is those unknown peaks are not addressed in the study. So you need for extractables, unknown extractable, you need to identify them to the extent that the toxicity assessment can be performed to qualify them, or you need to do a leachable study or simulation leachable to demonstrate that they do not leach to the drug product solution. So for leachable study, so the simulated leachable, you must identify the leachables at or above the AET level to the extent that the toxicity assessment can be performed to qualify them.
Great. Well, thank you. So thank you,
Thanks.
Xiaochun, for giving that very thorough overview there. So final words then. So we're going to reverse order. So Derek, anything you'd like to add before we close up today?
No, I think that's pretty good, a comprehensive overview. I think Xiaochun and Matt and then my information as well kind of covers the overview of the, not only the risk assessment piece, that's a first stage, but also then the analytical testing aspects and some of the challenges associated with it. So I think that's all I have to add in at this point.
And Matt, anything you'd like to add?
No, I think that's as Derek pointed out, a pretty detailed overview from each different area of the topic.
Well, look, gentlemen, I appreciate you taking time out to talk to me today. It is a really interesting topic and a very important topic, I should add as well. So, viewers, if you'd like to learn more about how PPD, Patheon, and Thermo Fisher Scientific can help you with all things extractable leachables, explore the links above this video, and it'll take you links to the various companies and how they can help you in the different stages around ENL. And also, if you've got any questions for Xiaochun, Derek, or Matt, then feel free to put them in the comments below the video, or you can connect with them directly on LinkedIn and ask them questions through that way as well. So Xiaochun, Derek, Matt, thank you very much for taking time to talk to me today. I hope the sun shines wherever you are in North Carolina and in Wisconsin, and I hope it continues to shine here in Surrey, in London, South London.
Indeed.
And,
Yes.
and until next time, viewers, as always, stay well and stay safe. Bye-bye.